Good morning all of you. Welcome to my channel Botany Now. This is Jyotirmay Nanda. In the previous video we have discussed the various parts of the flower, then the structure of male reproductive unit, that is stamen, which is differentiated into anther and a filament. We knew that the anther has two lobes and each lobe contains two microsporangia. Then the types of anther, and in this video we'll start the internal structure of the anther. Here we'll take the transverse section of anther. Let's start. When we draw the transverse section of anther, we see this type of structure. We have already discussed the anther has two lobes, and these two lobes are connected by a sterile tissue called connective. So this is the first lobe and this is the second lobe, and these two lobes are connected by this sterile tissue which is called connective. Then each lobe consists of two microsporangia. So these are the two microsporangia which is present inside the first lobe, and this is also the two microsporangia present in the second lobe. Then the space between the microsporangia and the anther is called the anther wall. So this is the microsporangia and this is the anther. The space between the microsporangia and the anther is called the anther wall. This anther wall consists of four layers of cells. What are those cells? The outermost layer is made up of a single layer of flat cells. So these are the single layer of cells which are the flat cell like structure. These flat cells are called the epidermis. So the outermost wall is made up of epidermis, and the function of epidermis is only protective. Next to epidermis, there is also the presence of a single layer of cells which are radially arranged. So these are the single layer of cells and these cells are radially arranged. These cells are called the endothecium, and these endothelial cells are arranged in a radiating manner, and its function is it helps in the dehiscence of the anther. Next to endothecium, there is presence of 3 to 4 layer of parenchyma cells which are loosely arranged. It is the third layer, so it is called the middle layer, and it is composed of 3 to 4 layer of parenchymatous cell, and these cells are also loosely arranged. And the functions of these parenchymatous cells is also it helps in dehiscence of anther. So what do you mean by dehiscence? Dehiscence is the point in which the anther is going to rupture here. So this is the point of dehiscence of anther. This point is called stomium. In this point, the anther is dehisced. This dehiscence point or stomium is the delicate and soft part of the anther. So when anther matures, these three layers, that is the epidermis, endothecium and this parenchymatous layer, become fibrous and hard. So these put pressure on the delicate part of the anther. Then stomium ruptures and the microspores or the pollen grain which are developed inside the pollen sac, they are released out from the anther. Then comes to the innermost layer, and it is very special layer. We called it as tapetum. These tapetal cells are pyramid-like structure. These cells are pyramid-like structure. We called it as tapetal cell or the tapetum. These tapetal cell is present in multinucleated condition. How is this multinucleated condition formed? This multinucleated condition is formed by endomitosis. Endo means cell ro bhitare, inside the cell. ଯେତେବେଳେ cell ଭିତରେ mitotic division ହବ or karyokinetic division ହବ, karyokinesis means karyon means nucleus. So when nuclear division takes place inside the cell, multinucleated condition arises. So ଏଠି କ'ଣ ହବ ନା cell ଭିତରେ nucleus ର mitotic division ହବ. So ଆମେ ୟାକୁ କ'ଣ କହୁଛେ ନା endomitosis ବୋଲି କହୁଛେ. So ଯେତେବେଳେ cell ଭିତରେ endomitosis ହବ ସେଇଟା କ'ଣ ଦବ ନା multinucleated condition. So these tapetal cells are present in a multinucleated functions of tapetum. What are the functions of tapetum? First, it is a nutritive layer, means it provides nourishment to the developing microspore. Microspore ଯେତେବେଳେ formation ହେଇଥାଏ pollen sac ଭିତରେ ସେହି time ରେ nutrition provide करे tapetum. Then second, it secretes certain enzymes. So the tapetal cell produces certain enzyme called callage, which dissolves the callose thickening formed in the microspore tetrad. Inside the pollen sac, the pollen grains are formed. Before the pollen grains are formed, the microspore are held together by the callose substances. These callage enzyme dissolves the wall and separate this microspore tetrad. से जो microspore माने produces से माने tetrad form रे थांती, से जो microspore tetrad से माने callose substance द्वारा परस्पर से तो bind ହେଇକି ରହିଥାନ୍ତି. These callage enzyme, a callage enzyme callose thickening को dissolve करिकी each microspore माननको separate करिथाय. Then the third functions, it helps in the formation of pollen kit. So pollen kit formation does not take place in all types of flower, but in case of hibiscus, which is called China rose, the pollen kit formation takes place. By chance, when our skin comes in contact with the flower, we found a yellow powdery mass or a yellow powdery substances attached to our skin. So that is the pollen kit, and this pollen kit is formed by the tapetal cell or the tapetum, and this pollen kit is present in the form of powdery mass. Then comes to the fourth function. The cells of the tapetum produce U-based body. So what do you mean by U-based bodies? These cells appear in the cytoplasm of the tapetal cells during the spore wall formation. ଯେତେବେଳେ microspore wall formation ହେଇଥାଏ ସେହି time ରେ tapetal cell ର cytoplasm ରେ U-based cell or U-based bodies appear करे. They are present in the cells of the tapetum and help in the development of pollen grains. ସେମାନେ pollen grain ର development ରେ help करथांती. These U-based bodies are coated with sporopollenin, which makes the outermost layer of pollen grain. So the U-based bodies are coated with sporopollenin. The sporopollenin is the most resistant substances. Pollen grain ର outermost layer sporopollenin द्वारा तैयारी होता है. The outermost layer of pollen grain is made up of by sporopollenin. So the function of tapetum is also the production of U-based bodies. So the anther wall consists of 4 layers. First is the epidermal layer of the epidermis. Second is the endothecium. Third is the parenchymatous layer. And the innermost is the tapetal cell or the tapetum. Then what is inside the microsporangia? Inside the young anther, there are the cells which are tightly packed inside it. So this is the microsporangia, and inside this microsporangia, these are the cells which are tightly packed. So these cells are called the archisporial cell. So these archisporial cells help in the formation of microspore or the pollen grains. As the anther is young, so this is the anther. As the anther is young, we called this structure as microsporangium. But when anther grows, the microsporangium also grows. So we call it as the pollen sac. So when anther young, we call the microsporangium. When anther matures or it grows, we calling this microsporangium as the sac. In this video, we discussed the layers of anther and their functions, and also the function of tapetum, which is very important. In the next video, we'll start the process of microsporogenesis. For further video, please like and subscribe to my channel. The PDF of all my slides will be available at the end of. Please take a screenshot of this slide.